I don't, not now. This is part of my problem. I studied not just knotted, but a number of genes. Knotted, rough sheath, ligulus 3, ligulus 4, lax midrib, hairy sheath freight, rolled 1. All of these genes do the same thing. They're all dominant mutants of a normal gene. So they're either, most of them are what's called a neomorph. By turning on in the wrong place, they add a function that's otherwise not there. And what they do, they all do the same. They extend the sheath into the blade, making inappropriate blade sheath boundaries. And I did a lot of work in my lab on studying the basis of this. And I have made up something called the maturation schedule hypothesis that it makes me smile when I see people maybe once every 2 years refer to it as something that does fit their data. And it has to do with cells at some time in their development get a time point saying begin your maturation. And then on their very own, as if epigenetically programmed, they go through a maturation. And at some point along that maturation, if the signal goes off and says differentiate, they differentiate as to their schedule, where they are in the schedule. And if you retard the schedule, you will pull juvenile characters into the middle of an adult, surrounded by adult tissue. It's all in this juvenile adult. In annals, you have words like pedomorphosis and stuff like that. I really got into this. When you have a juvenile sector inside of adult tissue, I looked at the details of that line at the level of the scanning EM, and that line is cellular. The cells are inheriting something about an identity that is not transferred from cell to cell. It's locked into their nuclei, not shared. I concluded at approximately at some point, I concluded that I could go no further, at least in maize. My lab was really good at finding out what layer of leaf something acted in order to do anything. Kathy Barton's actually studied this gene. This is rolled 1. You've studied it properly. I figured out by doing these genetic mosaic experiments, sort of plant gynandromorph experiments, that if the rolled product was expressed in just the lower epidermis, it was enough to switch the entire little organ patch around. Switch it up backwards, but it didn't turn the vascular system over, it just switched the outsides. That's the kind of stuff I did. I love that kind of stuff. Couldn't make heads or tails of it in the end. In 1992, I was in Heidelberg, and I heard Graham Moore talk about the subject that he published on 2 years later from Mike Gale's lab. Mike Gale's now gone. It was grasses line up in a circle. And that was from comparative mapping. It turned out that if you use the right conserved genes, all the grasses had the same genes all lined up in the same order, and they were simply rearranged, and it occurred to me, and Bennettson and I published on this, that if you sequenced in one grass, and you did your knockouts in another grass, and you did your expression studies in another grass, it was all the same thing. It's all one big system. It all transfers. It's all one big ancestral gene. And that the real system is not the individual model systems, but the real systems are the clades. And I wanted so much to get out of maize. Partly, let's face it, Arabidopsis was eclipsing maize in power, and the talent was running to Arabidopsis, and not me. How could that be? And it's not, maize was no longer the best system. And also, that I thought that maize could maybe make a comeback within the grasses. And the grasses, because they're commodities, you don't have to stress that corn is actually chicken and pig food. You could say rather that it is one of a whole group of related grasses. and simply the one about which we know the most historically. But other grasses like Soteria or sorghum or rice, all of these are being sequenced or sequenced. Brachypodium, sequenced. All of these are really the same thing. They're all variations, and any difference, that's of special interest. So what was I going to do about it? Well, it took me 10 years to do anything about it. And thanks to a graduate student, Nick Klinsky, real smart. He didn't want to study this himself, but he thought I'd appreciate it. So he just showed me a gene in maize and a gene in rice that I really cared about, the ligulus 1 transcription factor gene, the squamosal binding protein. And he showed me that outside of it were these conserved sequences that were conserved just like exons. But introns and everything else was all randomized away. And the only thing left were the exons and these conserved non-coding sequences. And I went on, finally, I found something. I did a lot of searching, spent a lot of money, failing to be able to work on grasses. I did 2 sabbaticals at Q. I can key out almost any grass if I can see a flower or a ligule. I can key it out, but it didn't help me. I couldn't get where I wanted to go, and then all of a sudden, through conserved non-coding sequences, here's 2 mid genes, one in sorghum and one in maize. There are the exons. See how they're conserved? These are blast hits after 50 million years of evolution. You see all these monster amount of DNA next to them that's also conserved. The little purple bibs are the ones that are pipeline, all done by programmers. Tell us in automated fashion where they are, and then we look and confirm that in fact those are the real conserved non-coding sequences, or almost so. Sometimes they're a little off. Enough of that. So now, in the next 23 minutes, I'm going to give you a feeling for why I'm not really ready to spend much time on a retrospective. I will be, but I'm not ready yet. Tetraploides and similar macromutations. That word macromutation comes out of Richard Goldschmidt's vocabulary, a developmental geneticist. May nudge evolution in predictable directions. That is, the mutation may push evolution into somewhere it otherwise wouldn't go. This is precisely the kind of thinking that was excluded from what's called the modern synthesis, the Dobzhansky consensus of the 30s and 40s. This kind of thinking, called mutationism, was rejected. I'm for a reevaluation. That's me, reevaluate. Here's the guy, the hero of the last stand of mutationism, a battle he lost. Right here is something that if you do nothing, absolutely nothing, like you don't have your pencil on the page or anything, just jot down American Scientist 4084 to 98 and read it. It's a long time ago. You wouldn't guess that anything could still be fresh or written in 1953 about the intimate nature of the chromosome. Really? I don't think they knew about histones. There was no epigenetics. How could anybody say anything about the intimate nature of a chromosome? Well, Goldschmidt had a lot to say about the intimate nature of a chromosome. And if you cut him a little slack, I think you'll see that he wasn't always wrong. But he had a real point. So he's the guy who gave us hopeful monsters and thinks that evolution is going in bursts, and that certain saltations, big events, are as important as little fine-tuning events. But only the little events are in the modern synthesis, and he's a big event guy. And there's nothing quite as big as you'll see as that tetraploid. So this is plants as we know them today. And these are, this is actually not quite up to date. I'm about 2 months behind in those that are sequenced and analyzed as to where their tetraploids are. But I got pretty many of them, and when there's a starburst, there's a tetraploid. Note that there's a papaya in the order, in the same order Brassicales as Arabidopsis, with 2 tetraploids that separate the two. There is grape there, So grapes had no tetraploidies. Back here is a hexaploidy, kind of a tetraploidy, and then a tetraploidies related in this way. Here's a double tetraploidy up here at the base of the monocots, the pre-grass tetraploidy, sorghum tetraploidy maize.